Hi, ma'am. I have sent you the link just now. Also, yeah, yeah, ma'am. This the YouTube link which I have sent just now, ma'am. Hello, you are listening, ma'am. Just one. Okay, wait. Yeah. Hmm. I have sent you the link just now. Yeah, yeah. Ma'am, this is the link which I have sent you. Hello, you are listening. Yeah, ma'am, I have sent. The new link which I have sent, you have forwarded that link, correct, ma'am? Then, yeah, YouTube link just 2 minutes before, yeah, or yeah, 2 minutes before, yeah, yeah. So two link just now because I don't know. I am logging into the same, and after that I closed that window which I was using it for like 7:58. So then again I logged in, and then again. So I have shared new link zero. Yeah, 2 minutes, 3 minutes before. Yeah. Hmm. 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 Okay. No, no. Yeah. Hmm. Yeah. YouTube link just now I have sent. Yeah. Hmm. Huh. Okay, ma'am. Okay. Thanks. Hmm. Good morning, everyone. So, so today we are going to see another sets of practical, which we are supposed to complete according to the syllabus of the life science for engineers. The first practical in the list is bacterial population growth. Uh, so, as we all know that bacteria are prokaryotic organisms that most commonly replicate by the asexual process of binary fission. So, in this, what happens is that normally a single cell divides into two identical daughter cells. The The bacterial population growth, as the name suggests, represents the number of live cells in a bacterial population over a period of time. Basically, there are four distinct phases of the growth phase. The one shown here represents lag phase, and the one which shows an exponential increase shows the exponential phase, then the stationary phase, and then the death decline phase. So the four distinct phases of the growth phase or the growth population curve of the bacterial cells, namely lag, log, stationary, and death phase, are as follows. So what happens in the lag phase is that there is no increase in the number of living bacterial cells. That means the cells can survive that phase, but actually there is no increase in the number of living bacterial cells. In log phase or the exponential growth phase, there is exponential increase in number of living bacterial cells. In stationary phase, it's a kind of a plateau stage in which the number of living bacterial cells is almost stationary as compared to the exponential phase, because in this phase the rate of cell division and the death is the same. That means the number of living bacterial cells which are dividing is equal to the number of bacterial cells which are dying. In the death or the decline phase, there is a decrease in the number of living bacterial cells. Let's see in little bit more detail what are the different phases of bacterial growth phase and what are the salient features of the different phases of the bacterial population curve. In lag phase, what happens is that the cell increase in size or the cell prepares for division, but actually there is no cell division happening in this phase. In the exponential log phase, the metabolic activity of the bacterial population is very high, as DNA, RNA, cell wall components, and other substances necessary for growth are generated for division. It is in this phase that antibiotics, which is commonly prescribed when one gets sick, is mostly effective in this phase, because the components like the cell wall components, DNA and RNA are mostly synthesized in this phase. In stationary phase, where the number of living bacterial cell is equal to the number of dead cell, in this the metabolic activity of the bacterial population decreases. In this phase only the bacterial spore formation takes place or pathogenic bacteria forms virulent factors. In death phase, the number of living bacterial population decreases. In this population, the nutrient contents become less and the waste product increases. Waste product of bacterial cell population is normally carbon dioxide, which increases, and then the nutrients which are available for the bacterial cell population also decreases. In this condition, the cells die or they lies, as a result of which what happens is that they will spill their contents into the environment, which can be used by the other bacteria as nutrients. So this condition, only some of the spores are able to survive this death phase. But what happens is that in these spores, when will provided with suitable environmental condition, they can start fresh population. So there are certain factors which determines the bacterial growth population. These factors are namely oxygen, pH, temperature, and light source. All these factors positively influence the bacterial growth. That positively influence the bacterial growth means more is oxygen, more is pH, or ambient temperature. The temperature means ambient temperature means each bacteria requires a specific temperature for at which their growth is maximal. If the temperature condition is altered away from the ambient condition, the bacterial growth might hamper. Light influence. The light plays a positive role. Bacteria requires light for proper growth. So if light conditions are maintained, the bacterial growth will be maximum. So let's watch a video which will explain properly how to do a bacterial growth curve in the lab. Just 1 minute, we will upload the video and then see. So we saw how bacterial growth curve is done. So the salient features of these experiments are as follows. What we need to do is that we need to prepare a culture. Culture means some medium will be there in which microorganisms will be added. In this case, the example which was shown here is Staphylococcus aureus. So you will incubate a culture overnight so that the cells starts to grow and acclimatize to the condition. Once, so in the morning, next morning, what you will do is that you will prepare media, fresh media, and then you will inoculate some portion of the culture which you have inoculated previous night and incubated overnight, and then you will add it to the culture and you will let it grow, and you will take the reading subsequently for every 30 minutes, and then the reading and the time you will plot it on the x x graph paper, and graph paper time will be on the x-axis and the OD will be on the y-axis, and from there you will find a sigmoidal curve which will specifically show three different phases of growth phase: lag phase, log phase, and stationary phase. If you want to see actually the death phase, then you need to do the growth curve for longer period of time. Okay, so this is us all about different phases of the growth of bacterial cell population. So in summary, what we have seen for this practical is that bacterial population curve is nothing but the growth of bacterial cells over a period of time. Then there are four different phases of growth phase: lag, log, stationary, and decline phase. In lag phase, there is no actual increase in the number of living cells. In log phase, there is an exponential increase in the number of living phase bacterial cell. In stationary phase, then there will be no increase in the number of living cells because the number of living bacterial cell is equal to the number of bacterial cells which are dying. In death or decline phase, there will be actual decrease in the number of living bacterial cells. And we also studied what are the factors which influence the growth of bacterial cell. The factors were oxygen, temperature, pH and light. Out of this, pH and temperature plays a very important role because for each bacterial species, there is a definite pH at which they will grow maximally. Similarly, the temperature requirement is also there because for each bacterial species, there is an ambient temperature at which the growth will be maximum. Alteration of temperature above or below the ambient temperature results in reduced growth. The other important features of the bacterial population was that in lag phase, the metabolic activity is low. In log phase or exponential phase, the metabolic activity is very high. It is this phase at which the antibiotics which acts against the microorganism are most active. In stationary phase, again, the metabolic activity is low. In stationary phase, bacteria starts to produce spores because this is an extreme condition, and the virulent bacteria starts to produce virulent vectors. In decline phase, bacterial cell starts to lyse and all their internal contents are released in the surrounding medium, which is used as a nutrient by other bacterial cells to survive. In this death or decline phase, only spores forming bacteria are able to survive, and when favorable environment arrives, these spores can again starts to grow. So now we will study another practical or which we are supposed to do. Its name is nitrogen cycle. Nitrogen cycle is something which is commonly observed in the outside environment in which we stay, and it's a daily phenomenon, and it's an important cycle which happens in the atmosphere. So what is the importance of nitrogen? Nitrogen is an essential component of all living organisms, and it is used for the synthesis of protein, nucleic acid, and enzyme. We have studied in our theory classes what are the importance of proteins, nucleic acid, and enzyme. Proteins are the functional unit of the cell. Nucleic acid is the genetic material of the cell, and enzymes perform specific actions inside the body. So all three are very important, and for all three, nitrogen is an important component. So we can see how important nitrogen is for living organisms. So the next question which we can ask is, what will happen if there is no nitrogen in the atmosphere? So when there will be no nitrogen in the atmosphere, this will lead to reduction in plant growth, as well as it will lead to reduced growth of the root. At the same time, when there will be less nitrogen in the atmosphere, the older leaves of the plant will turn yellow and they will start falling from the plant. At the same time, the crop production will be lowered when there will be less nitrogen in the atmosphere, and the plant plant along with the root system which are not developing well due to the less nitrogen in the atmosphere will also hamper soil microorganisms which are there, resulting in reduced growth or reduced proliferation of soil microorganisms. So when there is no nitrogen in the atmosphere, it impacts almost everybody, like if, starting from plant, resulting in reduced growth, and root, reduced stunted growth, resulting in yellowing of older leaves of plant, reduced crop production, as well as reduced growth of microorganism and their life of soil microorganisms which are dependent on the atmospheric nitrogen will be at stake. Now, what is nitrogen cycle? Nitrogen cycle is nothing but the converse movement of atmospheric nitrogen to the soil and again from the soil to the atmospheric nitrogen. This cycle is known as nitrogen cycle. From the atmosphere to the soil and from the soil again back to the atmosphere is known as nitrogen cycle. So nitrogen cycle consists of various steps. So these steps are namely nitrogen fixation, nitrogen assimilation, ammonification, nitrification, and denitrification. We will see each of these steps one by one. So nitrogen fixation in so that amount of atmospheric nitrogen is around 78%, but plants cannot use this nitrogen directly in the form of nitrogen. This nitrogen needs to be converted into ammonia before plants can use them. So the first step is the nitrogen fixation, in which the atmospheric nitrogen is converted is used by some of the plants, leguminous plant, which can absorb the atmospheric nitrogen in the ammonia form. Example is groundnut plant. Besides leguminous plant, which can take the atmospheric nitrogen and fix it in ammonia, there are some bacteria also which can fix the nitrogen. These bacteria are known as nitrogen fixation bacteria, and the examples of these bacteria include Clostridium, Azotobacter, Nostoc, and Anabaena. After atmospheric nitrogen is fixed in the form of ammonia, this nitrogen needs to be assimilated. Assimilated means the nitrogen in the form of ammonia or the nitrogen in the form of nitrogen which is present in the soil is first taken by the plant in the form of organic nitrogen. This plant is in turn eaten by animals which take plants as their food, as a result of which the organic nitrogen which was present in the plant from nitrate and ammonia present in the soil is taken by the animals, and now this organic nitrogen is present in the animal. So this process is known as nitrogen assimilation. Now the third step in the nitrogen cycle was ammonification. So what is ammonification? What happens in ammonification is that when animals die, the organic nitrogen present in the animal is converted into ammonia. And this ammonia, because these animals get decomposed when they die, as a result of which the organic nitrogen present in them is converted into ammonia. This ammonification is also being carried out by microorganisms called fungi and bacteria. So when animals die, their organic nitrogen content is acted upon by microorganisms called fungi and bacteria, and they will decompose the organic nitrogen, as a result of which ammonia will be formed. This can be easily seen in the environment. Sometimes you see that when some animal dies or when vegetables or fruits are left in the field, these fruits and vegetable as well as the animals are acted up, dead animals are acted upon by saprophytic bacteria and fungi, and they will be decomposed, and then ammonia will be formed from them. Ammonia will be formed from them. Ammonia is also produced from volcanic eruption and excretory product of animal. We have studied in our theory class there are some animals which also excrete ammonia in their excretory products. The fourth step in the nitrogen cycle is nitrification. In this step, what happens is that ammonia is converted into nitrate. So ammonia first is converted into nitrite, and this is being accomplished with the help of nitrifying bacteria, which is known as some of the examples which are for this type of bacteria are Nitrosomonas or Nitrococcus. This type of microorganism convert ammonia into nitrites. Then this nitrite is again converted back into nitrates. So this conversion of nitrite to nitrate is done by a group of microorganisms which are known as nitrobacter. They can convert nitrite to nitrate. So these three microorganism names are very important, and we need to remember that Nitrosomonas or Nitrococcus will convert ammonia to nitrite, and Nitrobacter will convert nitrite to nitrates. Then the last step is denitrification. Denitrification, what happens is that the nitrates which was formed from ammonia due to nitrifying bacteria is converted back into nitrogen, and which will be released into the atmosphere. This nitrate is first converted into nitric oxide, and then nitric oxide is converted back into nitrogen. And the function of converting nitrates to atmospheric nitrogen is being done by group of bacteria which are known as denitrifying bacteria. And some of the examples of denitrifying bacteria are Thiobacillus denitrificans, Pseudomonas denitrificans, and Micrococcus denitrificans. So now this nitrogen can be released back into the atmosphere. Now consider a scenario when there will be too much nitrogen into the atmosphere. So in that condition, what will happen is that too much nitrogen will lead to the formation of acid rain. Acid rain formation will lead to the acidification of lakes and soil, as a result of which there will be increase in the death of plants. So you can see neither too much of nitrogen in the atmosphere is good, nor too less concentration of nitrogen in the atmosphere is good. So there should be an optimum balance, or the concentration of nitrogen in the atmosphere needs to be maintained. And for this, nitrogen cycle plays a very important role. So in nitrogen cycle, what all we studied? In nitrogen cycle, we studied what is the importance of nitrogen. The importance of nitrogen is that it plays a very important role in the formation of proteins, nucleic acid, and enzymes. Then we studied what are the problems if there is no nitrogen in the atmosphere. It will lead to stunted growth of plant and root system. It will also affect the crop production. It will also affect the life of soil microorganism. Then we studied what are the different steps in nitrogen cycle. The different steps of nitrogen cycle are nitrogen assimilation. Let's see once more what all steps are. Nitrogen fixation first, then nitrogen assimilation, then ammonification, then nitrification, and then denitrification. In nitrogen fixation, nitrogen is fixed from the atmosphere. Then in nitrogen assimilation, atmospheric nitrogen in the form of nitrogen or ammonia is taken over by the plant, and this plant is again taken over by animals. Okay, as a result of which now animals will have organic nitrogen. Then ammonification, when these animals die, organic nitrogen is converted into ammonia. This is being done by saprophytic bacteria and fungi. Then in nitrification, nitrifying bacteria converts ammonia into nitrates. And then denitrification, nitrate is converted back into atmospheric nitrogen. and this is being played. This role is being played by denitrifying bacteria. Some of the examples which we saw: Thiobacillus denitrificans, Pseudomonas denitrificans, and Micrococcus denitrificans. And then we also saw the problems which will happen when there will be increase in nitrogen concentration in the atmosphere, which will lead to the formation of acid rain, acidification of lakes and soil, which will ultimately lead to increase in the death of plants. So the third practical which we were supposed to do is species interaction. So what do you mean by species interaction? Species interactions describe the relationship among organisms of different species living in the same location. What does it mean? Take an example there of predation. So species interactions can be of several types. One of the type is predation. In predation, what happens is an individual of one species, example lion, tiger, called the predator, eats another species called the prey. Example can be rabbit, cat, dog. So there is a relationship between the predator and prey. If we will see, carefully that predator prey cycle, then you can see in the beginning when there is high number of prey, that means when there is high number of predator, there will be less number of prey, or when there will be high number of prey, there will be less number of predator. But as the number of prey increases, example take the rabbit, when the number of prey or rabbit increases, the number of predator decreases. Okay, but once a prey reach a certain, means their number keeps on increasing, a time will come then predator also will start to increase. The number of predator will also start to increase, because now there is more prey available in the environment, as a result of which predator will also increase. Number of predator will also increase. But then as the number of predators will increase, the number of prey will again start to decrease. Now, once the number of prey will start to decrease, this will lead to the decrease in the number of predator, because now there is no food available for the predator. So their number also will start to decrease. Once the predator number starts to decrease, the number of prey slowly will again starts to increase. So there is a constant cycle between predator and prey numbers, and you can see how they are interconnected, because more the number of, so in the beginning there is more number of prey, so there will be less decrease in the number of predator. Once the prey number increases, predator number will also increase. Predator number increase will result in the decrease of prey number, and then ultimately prey number decrease will result in the number of decrease in the predator, and decrease in the predator number will result in the increase in the number of prey. So predator prey cycle is very important. And then the other type of species interaction is interspecific competition. What is interspecific competition? It is a type of competition between different species for the same requirement. The requirement can be for the food, the requirement can be for the space, same space. Take the example shown here. So the example shown here consists of two different types of species of bird. The one bird you can see in blue in color, and the one bird red in color. Okay, and both these type of birds are occupying the tree, same tree, for their survival. So this will lead to a situation where in which one species might get completely eliminated from a community because of the competition of the same limited resource, because both the different type of species are dependent on the same tree for their survival. So out of the two, the one which is better at utilizing the resource will survive, and the other which doesn't have any other option to survive will be eliminated from the community. This principle is known as competitive exclusion principle. So, in order to survive in that condition, what needs to be done is that resource partitioning. Resource partitioning is nothing but division of resources between the different species. It can be more clearly understood from the example shown here. Now, the two different types of birds, different species of birds, which were now can be seen that they are occupying the different regions of the same tree. The bird shown in red in color is occupying the top portion of the tree, and they are using the resources from the top portion of the tree. And the bird of another species, which is shown in blue color, is occupying the lower portion of the tree, and they are using the resources from the lower portion of the tree, as a result of which both species can survive on the same resource. This is called resource partitioning. Now, the other type of species interaction is called intra-specific competition. In this, the competition happens between the same species. In interspecific, the competition was happening between different species. In intra-specific, the competition is happening between the same species. Take the example of eagle. So eagle, they have the same so between the members of eagle, they are dependent on the similar type of food. Okay, so one eagle will eat one food, the another eagle also will go and eat the same type of food, and all these are the food of the same eagle. Okay, so now once all the members of the same species eagle starts to eat the same type of food, a time will come that the food source will become limited, as a result of which a competition will happen between the members of the same species, and this competition is known as interspecific competition. You can clearly see from the pictures depicted here that in the beginning 3 eagles and 6 food resources are available. Now 3 eagles, so first they will eat one food resource. So now 3 eagles, only 5. Then another will eat one another food resource. Then 3 eagle, 4 food resource. Now there is 3 eagle and there is only 3 food resource. So a competition will arise as the time goes by. So this competition is known as intra-specific competition. Now the other type of species interaction is symbiosis. So symbiosis is interaction between two different organisms living in close physical association, and they are of 3 different types: mutualism, commensalism, and parasitism. We will see what is commensalism, what is mutualism, and parasitism. In this list, the first is mutualism. You can see pollen, so insects living in close association with plant. Okay, this type of relationship is known as mutualism, in which both organisms derive some benefit. Like here the plant is there, so the pollinating insect will help in their pollination. In turn, the insect is getting food from the plant, so both the insect and the plant is getting benefit. So this type of relation is known as mutualism. Then the second type of symbiosis is commensalism, In this, one organism benefits, the other organism is neither helped nor harmed. Take the example of birds which accompany buffalo or cow in the grazing field. What happens is that when buffalo or cow, they walk over the grazing field, they will disturb the grasses or the soil which is there, as a result of which the or insects or small insects or big little insects, like example lizard, snakes, which are there in the surrounding and which is using grasses as a hiding place. So because of the movement of the buffalo or the grazing animals, their habitat will be disturbed and they will be coming out of their habitat, as a result of which these birds which is looking for the opportunity to for the small insects to come out of their habitat, they will thrive on that condition and they will make them as their food, as a result of which here the buffaloes is not getting any benefit, but because of the movement of the buffalo on the field, the insects which are coming out are being eaten by these birds. So this is type of a relationship which is known as commensalism, in which the buffalo is not getting benefit but the bird is getting benefit because of the physical association between the two, buffalo and the bird. Now the third type of symbiosis is called parasitism. In this, one individual is harmed while the other individual gets benefit. Okay, so take the example of tapeworm living inside a small intestine of human being. So what happens here? Tapeworm gets the benefit in the sense that it thrives on the digested food of the human being, but the human beings are not getting any benefit out of that. But in turn, what happens is that the food energy which was supposed to be used for the growth of the human being is now being used by the tapeworm for their survival and their growth, as a result of which what will happen is that human beings sometimes gets infected with some diseases and their growth is hampered, and this type of relationship is known as parasitism, where the human being is getting affected due to the tapeworm residing inside the small intestine. So in summary, if we look the three type of symbiosis, in mutualism, both the organisms are getting benefit. In commensalism, only one is getting benefit while the other is getting no benefit or no harm also. In parasitism, one is getting benefit, but the other is getting harm, like the example of tapeworm living inside the intestine of human being. So now, the sixth practical which we were supposed to do is Mendel's law. So Mendel's law we have already discussed in our theory classes, but let's give a brief revision of the Mendel's law. So Mendel's, as we all know, is known as the father of genetics, and he worked on pea plant, and his work on pea plant leads to the discovery of several laws which are known as Mendel's law. So Mendel's did two very important experiments. One of the experiment which he did was known as monohybrid cross. In that monohybrid cross, what Mendel did is that he took two pea plants of opposite trait. Opposite trait means opposite phenotype, like one tall pea plant and one short pea plant, and crossed them. Okay, and he found that all the plants which are generated from the crossing of tall plant and short plant are all tall in the F1 generation. F1 generation. Okay, so like we have generations called like grandfather, father, ourselves, then grandchildren. So like that also happens in plants also. So the first generation which will be coming out of the cross of one tall and short plant are all tall. Okay, then what he did is that again he crossed the plants which are all tall in the F1 generation to itself, and found that he obtained tall plant and short plants in the ratio of 3:1. 3:1. Okay, so supposedly if we got 4 plants, out of which 3 plants were tall and then 1 plant was short. But in F1 generation, all the plants were tall. Okay, 100% plants were tall. In case of F2 generation, which was developed after crossing of F1 plant, 75% of the plants were tall and 25% of the plants were short. This discovery led to the formulation of 2 laws, which is one law is known as law of segregation and the other law is known as law of dominance. In law of segregation, it is said that the offspring will inherit one trait from each parent. Like here you can see that these are all offspring. So the offspring, so tall is one trait and short is another trait. So now, so offspring, what they will do is that they will inherit one trait gene from each parent, like T from one parent and T from another parent. Okay, T from capital T from one parent and small t from another parent. Okay, so and the so this is called the law of segregation. So law of segregation is that each inherited trait is defined by a gene pair. Like tall, for tall it's TT, capital T, capital T. So each inherited trait is defined by a gene pair, and parental genes are randomly separated to the sex cells. So parental genes are randomly selected to the cell means this, if these are supposed to be gamete, like tall plant, okay, TT, capital T. So that will be randomly distributed into the gametes, like capital T in one and capital T in another. Then the plant for the another plant, like TT. So T is one, capital T in one and small t in another. Okay, so that is called. So parental genes are randomly separated to the sex cells, or so that the sex cells contain only one gene of the pair. Okay, so that means the offspring will contain one of these and one of these, or one of these, or one of these. So that is called the. So in case of human beings, like the child or the offspring will take one gene from father and one gene from mother. This is called the law of segregation. Now the law of dominance. So the law of dominance was that when Mendel crossed one tall pea plant and one short pea plant, all the F1 generation were tall. Okay, and this, when he crossed F1 generation plant, he found that 75% of the plants were tall and 25% of the plant were short. Okay, so this was because, and this, if you can see clearly, the phenotype, when T and both T are capital, the plant was tall. Okay, when one capital T is present along with one small t, again the plant is tall. That means the phenotype determined by this capital T is dominant over the small t. So this leads to the formulation of law, which is known as law of dominance. Because if you can see clearly, when there is no capital T, the plant is short. That means the phenotype of capital T is dominant over the phenotype of small t. In order for the phenotype of small t to be expressed, only small t should be present. Okay, if there is even a single capital T, that phenotype will be determined by the capital T, because the phenotype or the gene of capital T is dominant over the gene of small t, and this leads to the formulation of law, which is known as law of dominance. Now Mendel also did another type of experiment, which is known as dihybrid cross. In that, what he did is that he crossed two plants. One plant was like wrinkled and green, and the another plant was round and yellow. Okay, so here there are in the previous monohybrid cross only one phenotypic trait was considered, that is tall or short. But here two phenotypic traits are considered in case of round and yellow, and then one plant, and then the second plant the seeds were green and wrinkled. And when he crossed this plant in the F1 generation, all the plants were yellow and round, have yellow and round seeds. Okay, but when he crossed these yellow and round seeds of F1 generation, so if again the gametes, you all know about the law of segregation. Okay, so for law of segregation, out of this how many gametes will be produced? Okay, Y from one trait, yellow color, Y, capital Y, small y. Okay, then from here the other trait of shape, capital R and okay small r. Okay, so they will lead to the four different types of gamete. And when you could draw the Punnett square, Punnett square is nothing but the representation of the result of the dihybrid cross. So the Mendel found that in F2 generation he could find that out of the 16 plants, 9 plants were having yellow and round seeds. 1, 2, 3, 4, 5, 6, 7, 8, and 9 yellow and round seeds. And then 3 plants were having yellow and wrinkled seed. 1, 2, and 3. And then he found that 3 plants were round and green. 1, 2, and 3. And then one plant was green and wrinkled. So this experiment showed first two important things, that round, wrinkled and green is the recessive. That means in order for that phenotype to be expressed, everything needs to be present in homozygous condition. That means no capital Y or capital R should be there, because only small y will lead to the formation of green, and small r will lead to the formation of wrinkled shape. This experiment, he formulated the law of independent assortment. What does the law of independent assortment tells? That the genes for different traits are sorted separately. For example, round. So round genes will separate sort independent of the genes of wrinkled or yellow color. So the shape and the color, the genes will separate independent of each other. So this leads to the law of independent assortment. And the separation or the inheritance of shape or the color is not dependent on one another. They are independent of each other. So the second and the other important experiment which we were supposed to learn is gene mapping. This also we have studied in our theory classes. Gene mapping is nothing but the process of establishing the location of genes on the chromosomes. So it was, so gene mapping, as we have discussed in our theory classes, it was discovered by Alfred Sturtevant by working on the organism named as Drosophila melanogaster. And he used frequency of crossing over or recombination. Okay, so the frequency of crossing over or recombination was used for gene mapping, which is nothing but the mapping of the location of genes on the chromosome. For gene mapping, he used crossing over or recombination frequency. Okay, and crossing over or recombination happens during meiosis, which we have studied in our theory classes. Okay, so let's see how he did gene mapping. Okay, so understand gene mapping with an example. Suppose there is like gene A and gene B. Okay, And if the number of frequency or the crossing over or recombination frequency is 17%, okay, that means they will be located at certain distance on the chromosome. Supposedly, if this is with 17%. Now, if the frequency of recombination, okay, or frequency of crossing over is less, that means the distance between the two will be less, which can be clearly seen from the example shown here. So here the distance between the two, you can see here one and here one. If it is was 17%, they are located at a certain distance. If the frequency of recombination is 10, then they are located closer to each other. If the frequency of recombination is 39 or more than the previous recombination, then they are located at a greater distance. Okay. If the frequency of recombination is 50%, that means they are located on different chromosomes. So 50% recombination means the genes are not present on the same chromosome. They are present on different chromosomes. So by utilizing this type of thing, he mapped the genes location on the chromosome, and this was known as gene mapping. Understand this with more example. Okay, supposedly there are two genes, okay, VG and B, okay, and the frequency of recombination between them is 70%, 17%. Okay, so let's try to map this. Okay, so how this will be located? Supposedly this is VG gene and then this is B gene. We don't know. Okay, but we know that the distance between VG and B is 17% recombination frequency. Okay, so we map this as fine 17%, or the distance between them two is 17 centimorgan or map distance. Okay, because we know that there is a certain distance between this gene VG and gene B. Now there is another gene called CN, and the distance between VG and CN is 8%. Okay, now we need to find out the location of the gene CN. So now if the CN gene can be either located on the left hand side of VG or on the right hand side of VG. Okay, because the distance between the VG and CN and the frequency of recombination between VG and CN is 8%. Okay, so that means VG and CN should be closer to each other. So it can be either on the left side of CN or on the right hand side of CN. Now left hand side of VG or left hand side of right hand side of VG. Now there is another data available which says that frequency of recombination between C and B is 9%. That means the distance between C and B should be 9 centimorgan or 9 map distance. So that clearly shows that the CN location will be on the right hand side, because supposedly if you think that CN location should be on the left hand side, then if it is here, then the distance between CN and BN cannot be 9% frequency, because or 9 centimorgan, because this distance is clearly larger than 17%. So it should be somewhere between VG and B. So by using this recombination frequency, gene maps was developed. Take another example. Supposedly, the frequency of recombination between two genes A and B is 30%. Fine. So we will draw the gene location as A and B, and the distance between them is 30. Okay, fine. 30 map distance or centimorgan. Now, the another frequency of recombination between B and C is 45%. Okay, so B and C, the recombination will be B and C. The recombination will be 45%. Okay, now while drawing gene map, the first thing one should take into account is that before drawing G map, they should always look for the frequency of recombination starting from the beginning, because and the frequency of recombination cannot be more than 50%, because once it is 50%, they will be located on the different chromosome. So now out of this list, you can see that the maximum frequency percent is 45. So you will start with the first thing is that B and C. So B and C, you will draw B and C. The maximum distance between them will be 45 centimorgan. So this might be the distance between B and C. Now you need to find out the location of A and D gene. Okay. So, but now then you look for the next. So the next is the distance between B and D. So B and D is around 40% frequency of recombination. The distance between B and C was 45. B and C was 45. That clearly shows that the D will be located here on. It cannot be located on the other side because that will be on different chromosomes. Now, and the other thing is that D should be nearer to B as compared to C, because the frequency of recombination between B and C is 45 and the frequency of recombination between B and D is 40% only. Okay. Now, so once you have determined this location, now you need to find out the location of A. So, but now you saw that the frequency of recombination between A and D is 25%. Okay. So, and the other thing is that you also know that the frequency of recombination between A and B is 30. So now it should be somewhere between B and D. That clearly shows that the location of A is here. By using frequency of recombination, one can map the gene map on the chromosome. One can actually map, and it was clearly shown that the map, genetic map, which was developed was still holds true. But nowadays people use DNA sequencing by which you can find out exact location of each and every gene. But after that also the genetic map was found to be true. Okay. The last practical which we are supposed to learn is gene interaction. Gene interaction, as the name suggests, is the interaction between two genes. Okay. So this was, so one type of example where gene interaction is clearly shown is epistasis. And we have studied epistasis in our theory classes, where epistasis means that the effect of one gene is dependent on another gene. That is known as epistasis. Take the example of hair color. The genes, you might have known that some people might have hair color white, hair color green or red color. Okay. So the gene for, take the example of hair color. So the gene for hair color are some genes. Okay. So they might regulate the color of the hair. Okay. But take another scenario. So some people have hair on their head and some people don't have. They are called bald people. So in case of bald people, there will be no role for the gene for the hair color. That means the baldness gene is repressing the effect of the hair color. Okay. This means that the baldness gene is epistatic over over the gene for the hair color. Okay. And so the phenotype of hair color is dependent on the gene of the baldness. Okay. If baldness gene is not present, then only the gene for the hair color will be expressed. When the gene for baldness is present, the gene for hair color will not be expressed. This is known as epistatic. That means here you can see the example of gene interaction. That means the two genes are interacting, or in simple word, the phenotype of one gene, specifically the phenotype of hair color, is dependent on the expression of the baldness gene. Take another example of two mice which are like black in color. The coat color is black. So if there is a cross between the two black mice, okay, so you will see that out of the 16, 25% of the mice which will getting birth from this two, father and mother, you they will be like with no color. How it is possible? It is possible only because the gene interactions, this in that means the genes are interacting with each other. In Mendel's law, Mendel did not consider that genes are interacting with each other, but due to discoveries made after Mendel, it was clearly shown that genes do interact with each other. And the example is shown here. If you carefully look at this, so if you can see that whenever there is two small c, okay, small c, there will be the mice will be albino. That means when two small c are present, either capital B is there or small b is there. There will be no coat color. Okay, there will be no coat color. So in order to have coat color, out of the two c, one should be at least capital. Okay, one should be at least capital, then only some color for the coat will be there. Okay, you can say the example. See, in the case of brown coat, you can see that one capital C is present, one capital C is present, or both capital C is present. But when both C is are small, the mice are albino. That means the pigment gene pigment gene is epistatic over coat gene. Okay, the coat color gene. Okay, coat color gene, like the baldness gene was epistatic over the hair color gene. Here the pigment gene is epistatic over coat color. So this shows the example of gene interactions. So by this we are completing our practicals. So today we saw practicals related to bacterial growth curve. Bacterial growth curve is nothing but plotting of the number of cells over a specific period of time. Okay, so bacterial growth curve consists mainly, in the laboratory it consists mainly of three different phases. One is lag phase, log phase, and stationary phase. Theoretically, bacterial growth curve will have four different phases: lag phase, log phase, stationary phase, and death or the decline phase. In lag phase, there is minimal metabolic activity. The bacteria number doesn't increase, but the cell starts preparing for the division. Okay, in log phase or the exponential phase, the number of living bacterial cells increase exponentially. Okay, in stationary phase, the number of living bacterial cells doesn't increase. In death phase, the number of living bacterial cells actually starts to decrease. So in lag phase, the number of living bacterial cells doesn't increase. In exponential phase, or you can say like that, in lag phase, the number of living bacterial cells increase minimally. But in case of log phase, the number of bacterial cells increases exponentially. In case of stationary phase, the number of living bacterial cells doesn't increase, that it remains stagnant. In case of death or decline phase, the number of living bacterial cells actually starts to decrease. Okay, and then what we studied about was nitrogen cycle. Nitrogen cycle is nothing but the absorption of atmospheric nitrogen to the soil, bacteria, or the plants, and then again this nitrogen is again going back to the atmosphere. This cycle is known as nitrogen cycle. And nitrogen cycle consists of 5 different stages: nitrogen fixation, nitrogen assimilation, ammonification, nitrification, and denitrification. In nitrogen fixation, atmospheric nitrogen is fixed in the form of ammonia by leguminous plant, example groundnut plant, okay? Or it is also done by some of the nitrogen fixing bacteria. Then this nitrogen is assimilated in the form of nitrate or ammonia by plants in organic nitrogen. Okay, that means they will prepare ammonia, they will prepare ammonia, amino acid, proteins. So the protein products which they have made or the carbohydrate, this will be eaten by the animals. So organic nitrogen present in the plant is being taken over by animal. Okay, now this is called nitrogen assimilation. Then ammonification: when the animals or the plant die, the organic nitrogen is converted into ammonia. This is known as ammonification. This is done by saprophytic bacteria and fungi. Then comes nitrification, in which what happens is that ammonia is converted into nitrate. Okay, so nitrifying or nitrate. Okay, so then this is done by nitrifying bacteria. Then what happens is that this nitrate is again converted back to the atmospheric nitrogen. So this is done by denitrifying bacteria. Then we also saw the importance of optimum concentration of nitrogen in the atmosphere, which is 78%. Neither less nor good, neither less nor more is good for the living organism, because too low of nitrogen concentration in the atmosphere will lead to reduced growth of plant and animals, or too high concentration of nitrogen will also lead to reduced growth of plant and animals. Then we studied species interaction. Species interactions is the interactions between different or the same species, okay, and it is of different types. The first type of species interaction is we saw predator and prey. Predator is dependent on the prey for its survival because it uses prey as it is food. Example we saw of tiger and rabbit, or lion or rabbit. And there is a constant relationship between the predator and the prey numbers. Okay, when prey number will be low, predator number will also be low. But when prey number starts to increase, the predator number will also starts to increase. But once predator number increase too much, the prey number will automatically falls. But this fall in the number of prey will result also in the decrease of predator, because when there will be no prey, that means when there will be no food, there will be death of organism, which will lead to the less reduction in the number of predator. Once the number of predator falls drastically, the prey will start to increase. Again, when the prey number will increase, the predator will slowly increase. Then we studied the example of interspecies competition. Interspecies competition happens due to the use of the same resource by different species. This will lead to competition, which might lead to the elimination of one species. In order to prevent that, resource allocation needs to be done so that different species can use the different region of the resource. Example, we saw different birds occupying different region of the tree. Then we study intra-species competition. Intra-species competition is nothing but the competition between the members of the same species. Then we studied symbiosis. Symbiosis is the association between two organisms, okay, and this is due to the physical association between the two, and it can be of several types: symbiosis, mutualism, commensalism, and parasitism. In mutualism, both the organisms are getting benefit from each other. Example, pollen insects on plants. So pollen insects are getting food from the plant, and plant is getting help in pollination due to these insects. This is a type of mutualism species, mutualism and mutualism. In commensalism, which is an example where one partner is getting benefit, the other partner is not getting benefit. This is known as commensalism. In the example which we saw of birds staying in close association with grazing animals like buffalo or cow. Then the third type of symbiosis which we saw was parasitism. Example which we saw was tapeworm residing inside the small intestine of human being. Okay, in parasitism, one partner is getting benefit, but the other partner is actually getting harm. That is known as parasitism. Then we studied Mendel law, law of dominance, law of independent assortment, and then law of segregation. Law of segregation or law of dominance was derived from monohybrid cross. Okay, where only one phenotypic trait was considered. Example, tall or short. Dihybrid cross or dihybrid cross experiment led to the development of law of independent assortment. Okay, in this dihybrid cross, two phenotypic traits were considered, like shape and color. Okay, so and then we studied about gene mapping. Gene mapping was the mapping of genes on the chromosome, and for which the frequency of recombination was used. Okay, and then we saw how we can use the frequency of recombination for mapping the gene distance. Okay, then we studied about gene interaction. Gene interaction is nothing but the interaction between the two genes, and the example for gene interaction is epistasis. In epistasis, the phenotypic of one gene is dependent on the phenotype of another gene. Example, baldness gene is epistatic over the gene for the hair color. Similarly, the gene for the pigment is epistatic over the coat color. Okay, so these are the examples of the practicals which we were supposed to do. So most of this practical is like having a feel of the experiment. Okay, so feel of the experiment, like um, gene interactions, okay, gene mapping, or Mendel's law. This experiment cannot be done so easily in the laboratory. So you people need to have a feel. So, or the nitrogen cycle. So for that, mostly the experiments were discussed to you were of the theoretical nature. But the experiments like bacterial growth curve or sterilization, or the first experiment which we studied yesterday, or use of microscopy, and use of microscopy for the study of mitosis from the onion root tube. They are actually simple laboratory experiment which was demonstrated to you via online. Okay, so the exam pattern will be mostly of MCQ type, which you are supposed to give tomorrow, and the 50 questions will be there tomorrow and day after tomorrow. And out of the two, the best score from each, out of the two exams, the best score of the one exam will be used for evaluation out of the two. So any doubts or any questions are most welcome. I will send you people the slide. You can have a look, and then we will see how I can address the problem or in some way or the other. Okay. Thank you very much. And if you people have any doubt, feel free to contact me. I have already given you the email and my contact number. Okay. Thank you very much.